How big is the room? That's a good. So I could look it up. I'm going to say on the order of 4 meters, 5 meters. That's a good point, because when you look at this, you don't have a sense of scale. So the bats are small. I said they can't carry electronics earlier. I didn't tell you how much they weigh or how big they are. This species is about 15 grams typically. Grams. You could crush it in your hand. You shouldn't do that. But you can hold it in your hand. So in the experimental work, when you hold a bat, you can cup the entire bat in your hand fairly easily. And it's worth noting these are called big brown bats, as in they get smaller. By contrast, there are fruit bats, which are big. There's one called a flying fox, because it's about as big as a fox, but it flies. But they're not in the New England area or in America. So we can get the precise measurements of the room later, but for now, just about 4, 5 meters each direction. And so each of these chirps corresponds to some vocalization out in the array. Yeah, it's hard to get it to stop on one of the vocalizations here. There's more precise data than this. This is a video for show. But we know the precise vocalization times. We know how much energy was picked up at each of the microphones. And that's why we can get this gradient of color that you saw. So when you look there, you see different levels of darkness. The darker it is, the more energy there was in that direction. And the result is we can see where the bat's looking. Okay. That's a good question. It's so this is an interesting problem, and going back to, excuse me? Why did the bat go? Here. It'll be easier if I show the image as well. Why did the bat go through these 2 trees? More generally, why does it follow this path? Open question. There's a study that I referred to very briefly is one, and there's a whole group of studies with it, studying, the tracking of prey by the bat, evasive prey. So there was a collection of studies in this lab where there was a praying mantis let loose in the room, and the bat would fly after it and capture it. And it turns out you can model that pursuit behavior fairly elegantly in this, what's just called a prey pursuit law. I don't have any slides, and I don't have a whiteboard. We could talk about it after the talk. But so in some cases, you can model this behavior fairly well. By model it well, I mean you have some mathematical model of what's happening. It's a sync. Some kind of what's called a control law, something that could be implemented in a robotic platform, for example. In the military context, it would be missiles. I would like to think you can more efficiently deliver food packets for tracking evasive. I'm not sure why they'd be evasive, but so in some cases you can model the path, and you can explain it well in the sense that this model, this control law, yields some path on simulated data that correlates very well with the experimental data. In this case, this is a collection of data that hasn't been analyzed that much, but it could be. This was one of the first studies in which the bat, so with this particular setup of equipment, with this microphone array, one of the first where the bat's flying through a forest like this. And it's just waiting to be modeled, but we don't know yet why the bat chooses this particular path. This is a general problem in robotics, navigating through an obstacle field, the variety of solutions. Does anyone have that background, robotics, navigation? So, okay, I won't go into those for the sake of time, but okay, so we'll try to wrap up. It's an interesting question, and that's the nature of this work. When you're studying a biological system, you don't know, right? Without trying to go into religion, it's just this system there that exists, and we're trying to understand it. And you're trying to understand it, or I'm trying to understand it for its own sake, but I also think you can gain something. So in the case of that evasive law, that prey pursuit law, there's now a simple control law that can be used to manage some kind of aerial vehicle. So This array was limited. There's been some work to create what are called wideband arrays, ones that are not limited to 35 kilohertz, but cover a fairly large ultrasonic band. But they're limited because it's a difficult problem. It's a difficult problem because if you're going to record up to 100 kilohertz, by the requirements of the Nyquist rate, that means you need to be sampling at about 200 at least. 200 samples per second, per channel. What's more is if you have 10 to 16-bit sample width, that means you're generating a lot of data very quickly. And what's more is it's distributed around a room, a flight room, for example. And so my work was to pick up work by a postdoc in the lab to construct such an array. And we've had fairly good success. Since there isn't much time, I won't dwell on it for long. Essentially, they're little microcontrollers. It's a PIC family. It's a special one called the DS-PIC33F. It's called DS because it has some DSP hardware on board. That means it can do multiplication in a single clock cycle, and so on. These can be battery powered or powered by a wall adapter. There are 4 channels per board. They have SRAM built on so they can record at the high speeds required. They sample at about 260,000 samples per second. And the result is written to an SD card. So this is my attempt to address that problem of a wideband array. And we have 28 built. The system is designed to scale even more. It's made in such a way that you can put these nodes anywhere in the room. They can receive an external trigger, a TTL pulse it's called. Well, it is a TTL pulse. And that allows you to synchronize all of them. Yeah, I won't go into much more depth. But there's software associated with it for analyzing across array microphones. It's not done yet. I'm finishing up the project, and I'm going to try to make it open access, open source, freely available. Even if you're not doing ultrasonic work, I think this work involving a large array of microphones is interesting. Some of the code could be used, or the amplifier, or electronics design. So it's not open yet. It will be soon. Working on it. And so there's analysis software as well. Essentially, there's a bat. These magenta beams, or vectors, are scaled according to intensity in a particular band that you can select. Again, it's a wideband recording, which means you can freely focus on parts of the spectrum. And we get some sort of shape. This is an intensity plot with respect to azimuth and elevation. Essentially, at the center is where this target is. You might recall at the beginning of the presentation, I mentioned there was an experiment where the bat was trained to vocalize or attack a fake thing, a pole. This is that data. On the opposing wall was the array of microphones, an array of microphones. There were just 16 on the wall. And so here we see that the bat is actually vocalizing off to the side, not directly at the target. Again, the origin is at the center of the plot. And so we could talk more about that. One theory to explain that, or one hypothesis, is that when you have a beam of energy, if you focus it on something, on a target, it's easier to see if it's there. Recall that I said the more energy directed at something, the more easily you'll get a response back. If you have a beam directed off target, so here's a target, here's the beam axis, beam being the most intense direction. If you get this other part of the beam around the target, it's the case that for small changes in direction, small changes in degree or angle, there's a greater change in intensity of the returned echo, which means it's easier to localize something. By localize, I mean if my eyes are closed and I'm trying to find out where I'm snapping, localizing is finding that out, localizing the source. And so the idea here is simply that the bat is vocalizing off, away from the target, because it has sharper changes, steeper slope, however you want to think of it, off of it, off the main axis. So, and that's the summary, the end. In conclusion, biological systems are rich. You should consider them in your work. They're rich for inspiration for electronics. And I hope you are stepping away with some interest also in bat echolocation, nervous system, acoustics, but if not, certainly in the rich potential for ideas from the world around you. Thank you. And I don't think we have much time, but I welcome questions. If not, just find me anywhere after this.
This is normally the time when we'd be wrapping up. The keynote will be next, so there will probably be some movement in terms of screens, and they might want to simulcast in here. So if there's interruptions, there are interruptions, but I think until then, probably you can just take questions.
Okay.
Yeah.
So, and I can't see because there are bright lights in my eyes, but if you snap, that will be a sign.
Let's get the lights back on.
Oh, like a,
If you could repeat the question, because then the cameras will pick it up.
okay. So, yes? Yes. I know exactly what you mean. Yes. And so this study that I'm referring to here is a non-invasive. And you can get a lot of information when you have a rich array of microphones. Oh, I didn't repeat the question. Sorry. The question was, if this hardware is made more freely available, the hardware, software, and so on, analysis tools, could that reduce the extent to which invasive techniques are used? And the answer is yes. As non-invasive techniques become richer, simply put, there's no need to be invasive. There are ethical issues, and it's ethical, expense, regulatory issues that come up. So for this array, that could be an end that could help in that direction. This array represents about 2 years of work. And so making it freely available would certainly help in a variety of contexts, especially in the animal behavior community.
What are you using for microphones? Are you using MEMS?
I'm not. I'm using electret microphones. Does everyone... Oh, question again. So he asked, what microphones am I using? There's a company called Knowles. They produce microphones, especially those targeted at being implantable in your ear. There's a type of microphone called an electret microphone. If you don't know what it is, essentially one class of microphones can be thought of as a capacitor. So 2 plates. They have a particular charge on them, or a voltage rather. Well, okay, a charge. And when there's a sound coming, the plates move in accordance with the sound, which causes a modulation of the voltage signal. An electret microphone is one that has a charge fixed on it. Usually when you have this capacitor set up, you actually have to put a very large voltage across, as in about 150 volts or so. Yeah, so it doesn't work in this context. We use the small implantable in your ear microphones because it makes the array much smaller. So, okay, I'll even answer that a bit more than you might have wanted. The microcontrollers that I said have 4 microphones coming off. It's actually just a long wire. It's a shielded cable. It goes to a preamplifier board that has the microphone. It's very small. You can just stick it on a wall. I didn't go into it, but there are techniques for finding out where the microphones are in a room, arbitrary placement. So when they're small microphones, they invade the sound field less. As a last note on that wasn't asked for, but I didn't say this in the presentation, ultrasonic sounds, high-frequency sounds, are very directional. You can see this in the human hearing range. For example, when cars roll by and they have booming subwoofers, you can hear that everywhere. Annoyingly, maybe. But when you go up to the higher frequency, even in that sound, that song, the voices, they don't travel around walls. They don't invade your room. So that's, in the case of ultrasound, this microphone, the stem could block it. It had a fairly clear shadow acoustically, so.
Sorry, but you're
Yeah, okay. Thank you.